Good afternoon, students. So I'll be teaching you today about S1 nucleus. Since most of you, all of you have taken the biotics paper, so I have very few topics to teach you. So one of this topic is about transcript mapping, and it is about S1 nucleus mapping. So S1 nucleus is an endonuclease from the fungus Aspergillus oryzae, which cleaves single-stranded nucleic acid. So you can see here, there is a nucleic acid which is single-stranded, and you can see then it gets cleaved when you treat it with S1 nuclease. So it will cleave the single-stranded DNA or RNA. In this case, you'll see here this region is double-stranded. This region, which is there's a nick, this region is single-stranded. So when you treat it with S1 nuclease, it will cleave the single-stranded region, leaving only the double-stranded region. So S1 nuclease can be used for mapping, transcript mapping, which we'll discuss in the next slide. So here you can see you have an RNA. So RNA, it is hybridized to DNA, complementary DNA, but you'll see that this region, DNA consists of exons as well as introns. RNA, they have only exon region, but they don't have the intron region. So what happens? The intron, since they get paired with any of the RNA, they'll form this loops. So these loops represents the single-stranded region, single-stranded DNA region. So when you treat with S1 nuclease here, you'll see that the single-stranded loops are digested, leaving only the double-stranded region. And if you want to isolate the double-stranded region, you can treat with alkaline. So when you treat with alkaline, RNA is degraded, leaving only the double-stranded DNA. And this DNA represents the exon region because the intron regions have been already deleted. So only the exon region is present. So S1 nuclease is used mostly for start and end points of the transcript, which will tell you the start and the stop site of the transcript, or it can also be used for introns mapping, and this is known as transcript mapping. Transcript mapping will tell us where the start site are, where the stop site are, as well as if one, in this case, the principle is that if one of the strand is labeled at one end, the length of the label fragment remaining after hybridization and nucleus digestion reflects the point on the probe where two sequences diverge, and this is the basis for S1 mapping of transcriptional start site. So this one will explain in the next diagram how it happens. So in this diagram, we'll show you how it is used to locate the start point of a transcript. So first thing, you design a probe. This is the probe, you have designed it, and make sure the 5' is labeled. So you can unlabel it, the P32, using radioactive probe, or sometimes you can use fluorescent probe and the 5' in. You hybridize this one to a RNA. This is the RNA where you want to find the start site. So you hybridize them, and when you hybridize them, you make sure that this probe binds to the RNA, and part of it extend the 5' region. So this region will be single-stranded. So what you do, you digest with S1 nuclease, you digest it, and then you'll see that only the double-stranded region is left. The single-stranded region is left out, is digested. Then you find the size of this, say this fragment, on a denaturing pH gel, polychromate gel, and you'll see what's the size. See, if this one is say 1, 2, 3, 4. See, this is the full length probe. So this one is when there's no digestion, but when there's digestion, you're getting around say 400 base pair. That means the size from here to here is around 400 base pair. That means the start site is 400 base pair away from here. So you can find the start point by saying that from this region the probe is 400 base pair away. That means the start site is 400 base pair away. so you can find the location of the start point. So similarly in this diagram, so you can find the probe. So we design probe, label at the 5' region. Once you label the prime region, you digest it. Once you digest it, you find the size from here to here. See, in this case, again, see the probe is, this size is 300 base pair. That means from here to here is 300 base pair away the start site. Similarly, you can find the prime end. So label the probe here, digest it. Once you digest it, say the probe you're getting this after digestion, you're getting the size, say 200 base pair. So 200 base pair, that means the end side is 200 base pair from the region where the 3' end binds. So make sure when you find the start site, label the 5' end, and when you're labeling the 3' end, that means you are finding the end point. And make sure that this region must extend. This region must extend so that it should remain single stranded. So see in this diagram also, we'll see how to find the start site. So you have the RNA template. Once you have the RNA template, what you do is that you hybridize the probe. So this region will be the 5' region. Hybridize it. Once you hybridize it, then S1 nuclease. Then you run the DJ electrophoresis. Find the size from here to here. If the size is 100 base pair, that means the start site is 100 base pair from this region. From here to here, the start site is 100 base pair. Similarly, for the 3' end, if you want to find the 3' end, you can do the same thing, but label the 3' end. And same thing, you treat it as one nucleus and run the gel. And if you get the size from here to here, you find exactly how from here to here, from here to here, what is the size of the, how far is the 3' end of the exon or the gene of interest. So S1 mapping can also be used to map introns. So introns, what you do, you have the RNA which is the mature RNA or spliced RNA. This one doesn't have introns. You take the genomic DNA probe and label the 3' end, like in this case. So you label the 3' end. So this region, genomic DNA, consists both the exon as well as the intron. So this region represents the intron, which is single-stranded. So you digest it with S1 nucleus and see, you will get this one, the fragment. So in this case, you have labeled the 3' end. So we run the page, and when you run the page, you see the size. So in this case, we are getting the size of the fragment 50 base pair. That means the introns begins 50 base pair from the 3' end. So you can find the location of the intron. If you label from here, make sure that you find the size from here to here. And if you get 150 base pair, that means the size from here to here is 150 base pair. So I will show here also a diagram showing mapping of introns. So label the probe at one of the ends, hybridize to the RNA. Make sure this DNA probe contains DNA plus exons. So when you hybridize, this region represents the introns. So this region will be single-stranded, digest with the S1 nucleus, and you find the size of this probe. See in this diagram, you'll see that the probe says 1, 2, 3, say 300 base pair. That means the distance from here to here is 300 base pair. That means the introns is 300 base pair from the 5' end. So location, this one you can get in TA Brown. So in this case, what you do, here is SO3 fragments that contains 100 base pair of the coding region along the 300 base pair of the leader sequence preceding the gene has been cloned into M30 vector. So what you do is that you clone this gene. Okay, so you clone this gene, which is around 400 base pair. 100 base pair is the coding region, then is this 100 base pair, and 300 base pair is the non-coding region. And in this case, you have the SAU3A, so 3A restriction site, which it can cut at this region and this region. So you clone into M13 vector. So remember, M13 vector are used to get single-stranded molecule. So you get the single-stranded DNA. Then what you do, take this one and you hybridize it to a mRNA. So when you hybridize it, regions which are complementary will bind, okay, and regions which are not complementary, they'll come out. And you see that what you do is that you treat with S1 nuclease. So once you treat with S1 nuclease, the single-stranded region are digested, these regions are digested. Then you treat with alkaline to give you only the DNA. Find the size of the DNA. See, the size of the DNA you're getting is 150 base pair. That means the start site is 150 base pair from here. So you run this gel and you find the size 150 base pair. So the start site and this 150 base pair from the SAW3A site. So this is an experiment to show S1 mapping, how it's used. So you have the gene which has exon 1, exon 2 separate by introns, and the size is 7 kb, and it has restriction sign B, which stands for BAMH1, H stands for HIN3, and R stands for equal1. So these are the restriction sign. So you have genomic probe, so you can do two experiments, this one and this one. So this one is a genomic probe with BAMH1, and the size is total 7 kb. So what you do, you hybridize to the RNA fragment you want to study. So this is the transcript you want to study. So when you hybridize it, you'll see that you'll get this loop. So this loop represents the intron. So you treat with this one nucleus and there'll be digestion in the middle, resulting in a smaller fragment. That means two fragments. So you can see here, so when you use this probe, you get two fragments, one which is 2 kb and one which is 1.5 kb. So 1.5 kb and 2 kb fragments reveals that the introns is present in the center of the coding region. Similarly, you can use probe. There are three probes here. One is from BAMH1 to HIN3, which is 2 kb. Another probe from HIN3 to equal1, which is 3.5 kb. Another probe from equal1 to BAMH1, which is 1.5 kb. So you deal to the RNA fragment again in here. So this region you'll see again single-stranded, and you'll see that here also the RNA fragment, it has allele. So what you do is that you treat with an S1 nucleus, and you'll see that once you treat with S1 nucleus, you'll see here from here to here, actually this is 2 kb, but this region will get digested, and you get this region now represents a size of 1 kb. That means the start site is 1 kb from here, and this one represents what? This one represents the restriction enzyme, which is ENG. That means the start site is 1 kb from the HIN3. That means the start site will be 1 kb upstream of the HIN3. Similarly, in this case, the size is 1.5 kb. You do the S1 nucleus, this region which is single-stranded is digested, and the size which is left is 0.5 kb. And this region here represents the equal1 site. That means the end point is 500 nucleotides away from this R. That means 500 nucleotides downstream of the equal1 is the 3' end. Similarly, here also you get the fragment, and if you see the fragment, you are getting 1.5 kb and 0.5 kb, and that means the introns is from R, it is 1.5 kb, and from H the introns is 0.5 kb. So S1 mapping can be used for 5', 3', for finding the 5' start site or the 3' ends of a transcript, as well as for mapping of the introns. So this is all about S1 mapping. You can go through the books, T.A. Brown, from Gene Cloning by T.A. Brown. You can refer to that book, Gene Manipulation by Primrose. Yeah, you can get further details. So if you have any question, you can ask me in the comments. I'll answer you there. Okay, thank you.